Video for quiz 2, and this is very important quiz because it's just foundational. In the start of the course, I refer to this as a prerequisite information, and you know that lots of the stuff in the muscle differentiation videos you hadn't heard of before, but unfortunately, folks have heard, students have heard a fair amount about genes and DNA, and the unfortunate part is that we just got to get it so people are on the same page with understanding how well scientists understand genes. They're physical, they're made of DNA, they're composed of two parts. Output part, the transcription unit, that's going to be the output, that's going to be the part of the DNA that gets transcribed into RNA. RNA is the output molecule from DNA. And then there's the regulatory region, because we inherited all of these genes. We have them all in every one of cells. So which one are we going to use? When, where, and how much is what the regulatory region regulates for gene expression. And that regulatory region functions that way by having transcription factors bind to the regulatory region DNA and regulate the gene expression, regulate the transcription. That's why they're called transcription factors, because the function of transcription factors is to regulate transcription. We have learned about 3 transcription factors so far. MyoD is a transcription factor, myogenin is a transcription factor, and yesterday in class we learned some about the estrogen receptor, which is also a transcription factor. I always talk about the estrogen receptor. Of course, there's the testosterone receptor also in all vertebrate males, and the testosterone receptor is very much the same story as the estrogen receptor. So we could say that's 4 transcription factors that we've begun to learn about. We'll learn about several more in our course because they're the big switches in the activation of gene expression, which of course is a huge part of developmental biology. The timing and the location and the amount of gene expression is really what developmental biology is all about at its foundation. So this is foundational stuff that we just have to be solid on, and it's not going to change. It's just these are just straight up. This is the way this works, and this is the way that works. I've said that not all the scientists, not all biologists use quite the same terminology for the parts of the gene, and that's too bad because it doesn't help students. Even within developmental biology, you'll see, even in our textbook, they use a little bit different, and that can be confusing, and just ask me. What we want to do is be solid on at least one set of the descriptions, the organization of a gene, and if we hear something else, we can temporarily go, okay, I get that's why they're saying it that way. Okay, so what cells in your body? Another thing I want to emphasize is that I write these exams coming up in our course. They're going to have a lot of writing on them, and because we're going to learn a lot of stuff that's like story stuff that can be explained. And so we have to start getting mentally ready for being efficient on an exam. And so you can write more, but the answer to this question is just all. Woo, what happened there? Oh, yeah, it's just all of them. Which cells in your body have all of the myogenin gene? All of them. That's because of genomic equivalence. If you want to write that down in more words, all of them do because of genomic equivalence, you can. But if you wrote all, you would have to get full credit. And I'm not asking you to keep it short. When you know more things about something, it feels good to explain them in more detail. Sometimes on questions, I'll say, explain, and when I don't, you don't have to explain it, okay? There are always pretty short, direct answers that are complete answers in our course, and then there are longer answers where it takes more explanation. And that's because to learn the material, we talk about it, and we use examples and different settings and stuff like that. So it's just good to be aware of that, and it's good to ask questions in class, like about what's the answer to this question, or what's that, right? Which cells in your body have the myogenin mRNA? So the best answer we could give would be differentiating muscle cells. So before we dive into that details, it's really key to understand. We just established all cells have the DNA, not all cells have the myogenin mRNA. The myogenic mRNA is only present when the gene is being expressed, and it's always present when the gene is being expressed. So there's many, many copies of it in a cell that has the myogenic mRNA. And we can do many experiments to test that. Yep, we can detect the DNA, but does it have the mRNA? And that's when the gene is active. That's when it's playing its function. So just central principles to life on Earth. This is universally true for all cells on Earth. In bacteria, there's a little bit different terms for the RNA, but it's the same concept. Okay, and so myoblasts, that's not right. Those are muscle precursor cells that are not differentiating. And then if you just said muscle cells, I gave that credit. We haven't talked about it, but we haven't learned in our course whether the myogenin stays on, but I suspect it does. So differentiating muscle cells are a good answer. Myoblast is not a good answer. Okay, which of your cells in your body have the MyoD protein? So another really central concept. They have to be making the mRNA to have the protein, and if they're making the mRNA, there's a really good chance. It's unusual for the mRNA to be present and the protein not be present. So it's a 99% safe bet to say differentiating muscle cells. And you might feel that it seems like a trick question because the answers to these two questions are the same. But I'm testing that awareness of that if you aren't expressing the gene, you don't have the mRNA or the protein. It's not present physically. It's just a physical matter of fact. And like the example I often use is I say, what can we say about my head if my feet are in this room? And the answer is your head is in the room also. They're physically connected. Now, these are not quite physically connected. They're kind of indirectly connected. But it's like that. If you've got the mRNA, well, you almost certainly have the MyoD protein. If you have the MyoD protein, you definitely have the mRNA. Okay? And so again, right answer, wrong answers are the same. And if you have any questions, any confusion, even slight confusion of this, the first thing I do is to suggest you watch the videos again. And I encourage everybody to watch them again and just try to grind this stuff in and think about it and recognize how central it is. It's a specific example, but we're using it because it brings up a good universal, good setting to talk about the universal organization of DNA and genes and expression of genes on all cells, in all cells on Earth. So it's worth our time to learning. It's not just about humans, it's everything, and everything that's ever lived on Earth. So we're talking pretty big and bold. What is genomic equivalent? Genomic equivalent is each cell has the whole genome. Right? You can think about it. We're talking about the genomic. We're talking about the genome. And equivalent. What is equivalent? And the answer is each of the cells. They have an equivalent amount of the genome. And so there are lots of different ways to explain that. But the key concept is that each cell has the whole genome. In a case of sexually reproducing organisms, they have a whole genome from mom and a whole genome from dad, and it's very rare and only a few specialized cells in species that might not have the whole genome and not have the whole genomic equivalent. What part of a gene is the coding region located? And the answer is the transcription unit. Students struggle with this reality some, and it's a little bit curious, but there are two types of genes. There are just two types of genes. And one type of gene is called a coding gene, and it has a transcription unit, and it has a regulatory region made up of like a promoter and enhancer. And then another kind of gene, and this is true for all life on Earth, is called a non-coding gene. And non-coding genes make RNAs that are the final functional product. They make RNAs like tRNA, ribosomal RNA, small interfering RNA, and so on and so forth. RNAs that do not code for a protein, they're non-coding genes. The functional product, the functional output is the RNA. Coding genes code for proteins. Their output is also RNA. RNA appears to be the only output from any DNA on life on Earth, and it's called mRNA. For messenger RNA, it's going to bring a message to the protein to guide synthesis of that specific protein. Coding genes have a coding region that's part of the transcription unit. That's a coding region. That's where the codons, the triple letter nucleotides like AAU, that code for the amino acids. So coding genes always have it. The coding region is just a part of the longer transcription unit. That's fine. So it's part of the transcription unit and in coding genes like that. So, and importantly, do non-coding genes have a coding region? No. No, they're not doing that. They're making a product that's an RNA. These guys are making a product that is an RNA that will lead to protein synthesis. What is the function of all transcription factors? And the answer is to regulate transcription. They don't do the transcription. The RNA transcription is done. The RNA is produced by an enzyme called RNA polymerase. So it would be fair to say that transcription factors regulate transcription. You could also say they regulate RNA polymerase, but it's best just to say they regulate transcription. I think just kind of the broadest, truest definition that fits. Why do you think they're called transcription factors? Because they regulate transcription. Students can get confused with the difference between a transcription unit, which is made of DNA, and a transcription factor, which is made of protein. And they're both involved in transcription, right? So we just had to get comfortable with that reality. Transcription unit is the DNA. That's the basis for the transcription into RNA of a gene. The transcription unit is that region. And then a transcription factor is a protein that binds to the promoter or the enhancer and regulates transcription. Why do we need to have satellite stem cells? So lots of students get credit for this, but I always wanted to share the correct answer. The correct answer is because muscles are differentiated cells with limited growth potential. That's what's going on. That's what's going on. So we are in situations where we need more muscles. Healing, childhood growth, and working out are all things that lead to bigger muscles. So that's the best answer. There are other ways that say this that are fine, that were fine, that aren't as pinpoint. Most of the time they say, well, the satellite stem cells can make more muscles. Well, why would we need to make them? If we work out, and or heal, grow, that's why we need to make them. But also, we can't just make bigger muscles. The existing muscles can grow a little bit, just increase the muscle cell fiber size, but it's limited. And what is the output of DNA? And the answer is RNA. And so it's not proteins. The output is RNA, and I drew that out here. We have coding and non-coding genes in every case. For every gene, it's a transcription unit because that is the output. So it's just going to transcribe RNA, and in some genes, it goes on to code for a protein. And so a protein is a secondary output of DNA. And it's just important to appreciate because it's very simple organization of life on Earth. And I don't mean simple, easy to read. I mean easy to learn. I mean simple that it's the way life works. Yeah. And so I'm not aware of exceptions. There probably are some somewhere, but if there are, it's pretty interesting to hear about them. Give an example of a cell which the myogenic gene would be methylated. So this is a very easy question because it's really any differentiated cell that's not a muscle. So answering a muscle is not a great muscle. And I always say muscle, I should say striated muscle, because there would be some types of muscle that are not striated that would not need the myogenic gene. So for the level of our course learning, muscles, great. And so give an example of cell. So it could just be any other cell, liver, brain, lung, bone, almost any other cell that is a differentiated cell. If when they're the precursor, um, they would not have methylated. But after they differentiated, they would just, as far as we're concerned, maybe there'd be some exception to that, but as far as we're concerned, they would just methylate that myogenin gene. And the I was just trying to think about one thing. Oh, the best answer, I think, the most slam dunk answer would be, say, a chondrocyte. So that's a cartilage cell that's differentiated. Because that's a term we learned where we wouldn't have to say, a differentiated brain cell or anything like that. The best answer would just say a chondrocyte. And that's distinct from a chondroblast. The chondroblast is a precursor to cartilage, and a chondrocyte is differentiated. And we used it in the learning example, for the in the videos. So some students answer that, and that's just the most direct. Lots and lots of these are great answers. But if you think about it, that's the answer that you would be the solidest on, because we know it's methylated. We talk about it in the videos. It's methylated. That's why you can't turn it into muscle when you misexpress MyoD. What happens if MyoD is misexpressed in chondrocytes? And the answer is really nothing. That'd be a great answer. They're differentiated. They don't turn into muscles, because our understanding is that the myogenin gene has been methylated, and the transcription factor MyoD cannot overcome that DNA methylation, and general DNA methylation represses gene expression by repressing transcription. Yep. So then here, what's a gene? So we need to know this definition. Lots of students got credit when they gave a partial definition because we're just working on it, but we need to be solid on it. So we say physical. A gene is physical. It's physical, and it's composed of DNA. And it's just so important to say that, because we talked about it in class. For the field of genetics is still a relatively young field, 120 years old, and for almost the first half of it, they had no idea what a gene was, or very little idea what a gene was. And so they didn't think of it as being necessarily a really physical, simple thing like a DNA molecule. But we know that now, and we're proud to know that, and it's so significant in appreciating how that impacted the research that people were doing on embryology, because the embryo has all the genes. What genes is it using? Blah, blah, blah. So they're always made of DNA. And then in our course, in a developmental biology course, we'd like to go on and say that the gene has two parts, composed of DNA, organized in two parts. And we can add that too. If you didn't list the two parts, you got credit, but we know it, so Adam, right, that is a great answer. Transcription unit and regulatory region. And the last thing is we need to say it affects the phenotype. The gene is the fundamental unit of inheritance, heritability. And so it is that because it affects the phenotype. So when I ask this on an exam now, I would just you'd need to have all those: physical, DNA, two parts made of transcription unit, regulatory region, affecting the phenotype. So, you got credit, and this is a good example where I gave credit to a lot of students that did not have a complete answer. Okay, so what does MyoD protein bind to? So some students said enhancer, and that's wrong, because it should say enhancer of myogenin gene. There are lots of different transcription factors, and they have different target genes. We say the MyoD protein has target genes. One of them we're learning about is myogenin, and so myogenin is a MyoD target gene. And that means it doesn't bind to every enhancer. There are thousands of different enhancers and thousands of different transcription units, transcription factors. So we got to be specific, and it's just really important to appreciate that. Like if somebody said, "Where do you live?" even if you said an apartment, well, that's not a full answer. Right? Well, where? Which one? Like that, right? Because they're not going to find you if you just say apartment. And I don't mean to sound kind of scolding about this. I mean to say it kind of like it's just the way it is. This is just a matter of fact stuff, and you can learn it. We're not going to fudge the definition later and adjust it and bring in a lot of exceptional examples. We're just going to roll with these as just the fundamental thing. The hope is that we could roll in with this knowledge from our intro bio courses and our other biology coursework, but it just isn't get completed, and it's not as rigorous in some other settings. And so then these. So when we draw a gene, we say, well, that's the DNA, and these are things that are DNA. Transcription unit is, the promoter is, the enhancer is. These are things that are not DNA. The transcription factor, it's a protein. The RNA, it's RNA. So the answers to these are that the promoter is DNA, transcription factor is protein, and RNA polymerase, protein, enhancer DNA, transcription factor DNA, and mRNA is RNA. And you got one wrong for each of those if you got them wrong. And so I just have to emphasize again that if you feel kind of uncertain of any of these in this quiz, please go watch those videos again. And if you're pretty sure about them, please go watch them again, because there's a lot in those videos that we need to have as working knowledge, just daily working knowledge that you just walk into class with or you study with like that. Okay, awesome. Thank you. We're all set. If you have any questions about this, don't leave it, because if you're confused or concerned at this point, we got to make sure that we get this all ironed out and all set, for our course. Yay. All right, great. Thank you very much.